Welcome! Today, we are diving deep into one of the most incredible pieces of engineering in the universe. And no, it's not made of steel or silicon. We're talking about the molecular machinery that actually powers life itself.
So let's kick things off with a huge question, one that our source material really puts front and center. What if every single living thing, from the tiniest bacterium to the biggest blue whale, runs on a kind of perfect chemical software?
Scientists figure there are somewhere between 8 and 10 million species on our planet. That is a staggering amount of diversity. And yet, our source points out something incredible. They are all built from the very same foundational element, a flawless program for life.
And that program? Well, of course, it's DNA. And you should really think of it exactly like software. Its job is to do two things, and to do them magnificently well. First, store all the information needed to build and run an organism, and second, copy that information perfectly so it can be passed on from generation to generation.
Okay, to really get a feel for the scale of this, we need to zoom in, way in. So for this explainer, let's use a model organism, the common bacterium E. coli, and see just how this software is stored. An E. coli cell is tiny. Just 2 micrometers long. To give you some perspective, you could line up 500 of them, end to end, right across a single millimeter. It's almost unbelievably small.
Okay, but get ready for the truly mind-bending part. If you could somehow take the circular strand of DNA that's packed inside that tiny little cell and stretch it out into a perfectly straight line, it would be 1.7 millimeters long.
Yeah, just let that sink in for a second. The DNA, the software, is 850 times longer than the cell that has to contain it. How on earth is that even possible?
This is just a brilliant way to picture the engineering challenge here. It's not just crammed in there, it's perfectly organized, it's totally accessible, and somehow it never ever gets tangled. It is an absolute masterpiece of data storage.
And the information density? It's just as stunning. That single strand of DNA, built from just 4 chemical letters, holds the blueprint for 4,494 different genes. That's the informational equivalent of a 10,000-page book. Okay, so we've got this incredible library of information. But how does it get copied? Let's get into the process of replication. The source describes it as this highly disciplined orchestra, with a whole bunch of different parts working in absolutely perfect harmony.
Right off the bat, this process is anything but random. Out of 4.6 million chemical letters in the E. coli genome, the copying process always starts at one very specific 245-letter sequence. It's like having a single, unique start here address for the entire operation.
So let's meet the cast of molecular characters that make this all happen. You've got helicase, which basically acts like a zipper, unwinding the DNA. Then there's DNA polymerase 3. That's the master builder, adding thousands of letters a second. You've also got polymerase 1, which is the editor. Topoisomerase is the knot remover, relieving all the tension. And finally, DNA ligase comes in like the glue, joining all the fragments together. But the real star of the show has to be polymerase 3. The source describes it like a 3-armed giant. It actually copies both strands of the DNA helix at the same time, even though they run in opposite directions. It's just an unbelievably sophisticated piece of molecular machinery. So we know the process is fast, and it's incredibly complex. But what about accuracy? Well, this is where the story goes from just incredible to nearly unbelievable. Let's take a look at the quality control system. See, the polymerase enzyme doesn't just write, it reads what it just wrote. After it adds each new chemical letter, it immediately double-checks its own work. And if it finds a mistake, it actually backs up, cuts out the wrong letter, and puts the right one in before it moves on. So what's the end result of all this constant proofreading? Well, the final error rate is just astonishing. 1 mistake for every 10 billion letters copied. For all practical purposes, it is a perfect copying machine. This is why life can be so stable from one generation to the next.
Okay, let's pause here for a second. So far, we've explored the established science, the massive data storage, the complex machinery, the near-perfect accuracy. Science tells us how this all happens, but it doesn't necessarily tell us why. And that's where we're going to shift gears. From this point on, we're going to explain the philosophical conclusion that the author of our source material draws from all these scientific facts. The author kicks things off with a mathematical argument. They calculate that the probability of even a short 1,000-letter DNA sequence forming correctly by random chance is 1 in 10 to the power of 600. That's a 1 followed by 600 zeros. It's a number so huge, it's practically zero.
So, based on that staggering improbability, the author arrives at a pretty simple conclusion, which they state just like this: if there is writing, there is a writer. If there is perfect software, there is a programmer. And to support this view, the source highlights a quote from Professor Francis Collins, who was the head of the Human Genome Project. He famously described DNA not as just chemistry, but as a complex program that has the signature of a magnificent intelligence. The author then uses a very powerful analogy to really frame their argument. They say that believing this level of complexity just happened by chance is logically the same as believing that a tornado could rip through a junkyard and randomly assemble a fully functional Boeing 747. And so, this brings us to the ultimate conclusion presented in the source material, that this incredibly intricate, flawless molecular operation inside every living cell doesn't point to random chance, but to a creator with infinite knowledge and power. And with that, our look at this source material ends with one final provocative question that really gets to the heart of the author's entire argument. We all accept that even simple software needs a programmer. So the author asks, can the infinitely more complex software of life write itself?